Our next speaker is Chloé Loiseau. Chloé is working at the Swiss Tropical and Public Health Institute, and she does research on mycobacterial genomics, molecular evolution, and population genomics. The topic of her talk is ecology and evolution of multidrug-resistant mycobacterial tuberculosis. Thank you, Chloé.
Thank you. Hi, yes, thanks for the introduction. I work at the Swiss Tropical Public Health Institute in Basel. I'm a postdoc in the group of Sebastian Gagneux, and we work on tuberculosis, and today I'm gonna present some of our work on multi-drug-resistant mycobacterium tuberculosis. So, some key facts about TB. It's caused by, it's an ancient disease caused by a bacteria that transmits by microscopic droplets when a person coughs or sneezes. Of course, it disproportionately affects low and middle income countries, but it really has a worldwide distribution. You find it in every country. Every year, there are more than 10 million new TB cases, of which almost 500,000 are multi-drug resistant. And unfortunately, it has regained its infamous place as the top infectious disease killer after COVID, with 1.3 million deaths, at least in 2022. As I mentioned, TB is a very ancient disease. It has affected mankind for thousands of years. And despite this, we still rely on very ancient tools to control TB. We have a vaccine, BCG, which was developed in 1921. It's the only vaccine we have, and it offers partial protection. We have diagnostic tools which also date back to a century ago. Drugs which are often toxic can also be painfully injected, and clearly suboptimal monitoring. No fancy dashboards that are updated daily, more an annual report. And in many countries, it's still, paper-based forms used, and yeah, that's it. So what causes TB? It's a group of clonal bacteria called the Mycobacterium tuberculosis complex. There are 9 phylogenetic groupings that we call lineages. Some are adapted to humans, lineage 1 to lineage 9, and they're depicted here in the phylogenetic tree in these colored triangles, and some are adapted to mammal species. And the human adapted forms of the Mycobacterium tuberculosis complex shows phylogeographic distribution, which we see on the map. Some, are more widespread, some are more localized. And interestingly, they differ in some important clinical features, such as the rate at which they acquire drug resistance or transmit or progress to disease. For example, there's this lineage 2 family in blue, which has repeatedly been shown to be associated with all 3 of these features. So TB is curable, and the drug-sensitive forms of TB can be cured with a combination of 4 antibiotics, isoniazid, rifampicin, pyrazimide, and ethambutol for 6 months, and for a cure rate of around 85%. And if a patient develops resistance to isoniazid and rifampicin, then we talk about multi-drug-resistant TB. And then, of course, if a patient has MDR-TB, the therapeutic options are less evident. We rely on fluoroquinolones, aminoglycosides, and other drugs which are more expensive and more toxic, and have to be taken for a longer period of time. And there is some good news in the sense that for the first time in 50 years, there's been new therapeutic options that have come, such as bedaquiline and delaminid. And if patients then develop further resistance to one of these classes of drugs, we talk about pre-XDR or XDR-TB for extensively drug-resistant TB. And as I said, there's some hope with these new drugs, and this picture shows a bit the 1 day of typical XDR treatment, it's up to 2 years. And this is what the daily uptake for a patient. And now with these new regimens, clearly the pill burden has been really reduced, and that's good news in the field. However, MDR-TB does remain a public health crisis, since only 2 in 5 people with drug-resistant TB will even have access to treatment. So how does the bacteria develop resistance? It's a simple bug. There's no resistance plasmids. There's no evidence of horizontal gene transfer, which means that resistance arises through chromosomal mutations or small indels. And generally, the path from drug-sensitive to XDR-TB will be that first the bacteria will evolve resistance to isoniazid and then to rifampicin. And at this point, the bacteria is, the patient has MDR-TB. And then for the resistance to fluoroquinolone or the new drugs, and we talk about this pre-XDR, XDR-TB, which of course is associated with worse treatment outcomes. So there are 2 ways of acquiring drug-resistant TB. The first would be when there's a subpopulation of resistant bacteria that emerges within a patient and then is selected through treatment, through inadequate treatment on non-adherence to a regimen. And then the second scenario is that a patient that already has drug-resistant TB transmits to someone else. And of course, the burden of MDR-TB is a combination of both scenarios, acquired resistance and transmitted resistance. But a lot of studies now show that the majority of the burden is actually due to transmitted resistance rather than acquired resistance. And therefore, it's important to also understand what are the factors that drive transmission from one patient to the other. And some factors and well-known factors that influence the spread of MDR-TB is the environment in which a person is. So typically, environments like prisons or mines or townships, they're very overcrowded. They have high-risk individuals, and it's poorly ventilated areas, which favors transmission. Patient comorbidities can also affect, and then there's, of course, everything that has to do with the pathogen genetics. And one of the most important factors, actually, that explains, that shapes the rate and size of the epidemic is the fitness of the bacteria. So what is fitness? It would be defined as the ability to survive and reproduce. And in the context of infectious diseases, this means establishing an infection, replicating within the host, and then transmitting to a new host. So what do we know about fitness of drug-resistant strains? Well, generally resistance is accompanied by a reduction in resistance is accompanied by a reduction in bacterial fitness in the absence of antibiotics. And what do we know about it in TB? So we have in vitro studies that have shown that basically all laboratory-derived rifampicin-resistant strains were shown to have a fitness cost in vitro. And this is what this plot shows. We see different mutants so rifampicin-resistant mutants. These are different mutations in RPOB. RPOB encodes the RNA polymerase, and it's the target of rifampicin. And basically, we see that all the bars are lower than 1, which is the fitness of the susceptible ancestor. So that shows that regardless of the mutation, we see a fitness cost in vitro, although there are some mutations that have a higher fitness than others. Of course, bacteria evolve strategies to mitigate the effect of the cost of resistance, and they're able to evolve these secondary mutations called compensatory mutations. And these have been shown in vitro to overcome mitigate the cost of resistance. For rifampicin, for RPB mutations, resistance mutations, we find these compensatory mutations in RPO A, B, C, which are the 3 subunits of the RNA polymerase. So our research questions are that, so we see all this in vitro, and we basically want to know now in the clinic, does this also have an effect? Is it important? So is MDR-TB associated with the fitness cost in human populations? And of course, what is the role of compensatory mutations in humans, actually, not only in vitro? And why are these questions important for TB? So this is a map showing the proportion of MDR-TB cases amongst new cases. And globally, only 3.4% of new TB are actually MDR-TB. And this rate actually has been stable over decades, which could be surprising given that TB has been treated with antibiotics for more than 50 years. And it has also, this observation was also a bit used to say, okay, there is clearly a transmission cost. Resistance is not so much a big deal in TB, but what we see is that it's not, this relatively low rate conceals significant variation among countries. And we see that these countries in dark red, countries of the former Soviet Union, and they have much higher proportions of MDR-TB amongst new TB cases. And so these make a good clinical setting for our research questions. So we designed a study in the country of Georgia, the Republic of Georgia over here. We designed a population-based nationwide genomic epidemiological study. We sampled more than 90% of all TB cases, all drug-susceptible TB and all drug-resistant TB cases circulating in the country over 3 years. And to answer the questions of, is there a fitness cost in the clinic? And what is the role of compensatory mutations? And we sequenced all the genomes. There were around 4,600 mycobacterium tuberculosis genomes, and from it we were able to do genomic analysis, and for drug-resistant profiles, compensatory profiles, reconstruct transmission chains, and apply some phylogenetic models to compare the drug-susceptible and drug-resistant population. So the first thing we see is that this is a phylogenetic tree of the drug-susceptible population. So I split the drug-susceptible and the drug-resistant population. There are 3,000 genomes, and we see that there are 2 lineages that circulate in Georgia. We have lineage 2 here in blue and lineage 4 in red. And then if you look at the drug-resistant population, we see that the image is a bit opposite. And we clearly see that MDR-TB, so the drug-resistant population is associated with lineage 2. And then we applied some phylogenetic models. We collaborated with Tanya Stadler's group at ETHZ, who applied these models to compare the transmission rate of the drug-resistant population to the drug-susceptible population. And at first, we split by lineage, since we see this difference. And what we see is that in lineage 4, we clearly see a fitness cost. The transmission rate of the MDR population, here in yellow, is clearly lower than the drug-susceptible population. So far, so good. And then in lineage 2, however, we do not see this fitness cost, which shows that, okay, MDR fitness cost is lineage specific. Then we thought, okay, let's split this by the type of RPUB mutations. And for this, we split by drug resistance, by rifampicin, MDR strain that had an RPUB S450L mutation, and RPUB other mutations in S450L. And we see that MDR fitness cost also depends on the type of resistance mutations that the bacteria has. And RPUB S450L, which was also shown experimentally to have a lower fitness cost than other mutations, we also see this. It also translates in clinical fitness or transmission, and it has a very little fitness cost compared to the susceptible population here in gray. Whereas the lineage 2 MDR strains which have other RPUB mutations, they clearly have a fitness cost. In lineage 4, regardless of the resistance mutations, we see this fitness cost compared to the susceptible population. And then we thought, okay, now we want to know what is the effect of compensatory mutations. So we further split the lineage 2 the RPOBS for 50L between those that have a compensatory mutations and those that don't. And we see that MDR fitness cost depends also on having compensatory mutations or not. And actually that we have, so these are the group of strains which are lineage 2, which have the RPBS for 50L resistance mutation and compensatory mutation. And they are as fit as the susceptible, if not a bit more. And this was not the case for the RPOB strains that have other than S450L. So then clearly we have this in Georgia, we have this very successful, let's say, genotype that circulates, composed by these, genetic features. And we wanted to see, okay, how much of the MDR population is represented by this highly successful genotype. And actually, it drives transmission of MDR-TB in Georgia. And we find more than 60% of MDR strains which are this successful genotype. So this is the MDR population. And then we wanted to, given this finding, we wanted to look a bit deeper into the MDR population, and we had a retrospective data set that goes back 3 years. So we added this collection and we analyzed the 6 years of MDR TB from Georgia. This amounted to 1,600 genomes. And this is the phylogeny of the MDR population over 6 years. So it looks the same as before. And what we did is we looked at the transmission clusters in the MDR-TB population, and we saw that 65% of the population were found in transmission clusters. And although most of these clusters were only made of 2 strains, they were exceptionally high clusters. And I just want to highlight one, which is here in yellow. It was a pre-XDR cluster of 183 strains. Actually, I have some data that goes after 2016, and it just shows that it keeps on growing over time, which just shows uncontrolled transmission of resistant strains, and not only resistant strains, but highly resistant strains. Because if you look at the resistance profiles and the number of not just MDR, they have much higher levels of resistance. They carry a median of 6 drug resistance mutations, and of course, the more resistance you have, the less chance of treatment success you have, which is a problem. And so then for every cluster, and there were 212 of them, we reconstructed the transmission chains, which means that, for example, this is a cluster of 14 strains, and never mind the colors, but every node represents one sample, so that comes from one patient. And we're able to see, okay, this person transmitted to this person, this person, this person, that person. And then from this, we're able to infer the number of secondary cases for each case. And we use this metrics. We run logistic regression model using this as the number of secondary cases and and we look at the different bacterial and host factors that are independently associated with a high number of secondary cases. And we find, of course, that as expected, compensatory mutations and lineage both contribute to higher number of both associated with higher number of secondary cases. And then we also found that prison isolates, so patients that are incarcerated in prisons, was also associated with higher number of secondary cases, which is not so surprising given that prisons are associated with very overcrowded environments and people that often have also comorbidities. But then we also thought, okay, if prison is an environment that favors transmission, perhaps it's also an environment that could favor the evolution of compensatory mutations because of more transmission, more replication within hosts, more evolution of compensatory mutations. And in agreement with this, we did find that that prison isolates, that compensatory mutations were overrepresented in prisons. Yeah, and we then looked at. So I'm going back to this big cluster here. We reconstructed the transmission chain of this big cluster, which contained both prison isolates and civilian isolates. And the nodes are colored as a function of this. So purple nodes are prisoners, and the orange nodes are civilians, and the darker the shade, the more recent the sampling is. So the cluster starts here with, so that's the first case sample from 2011. It's a prison isolate, and if you follow the transmission chain, you see that we just visually, we go from purple to orange. So we go from prison isolates to civilian isolates, and we just see this spillover of MDR-TB in the general population, in the civilian population. And we did this exercise for all the clusters which contain both civilian and prison isolates, and so to basically quantify the spillover, and we see that over years we do see that we see more and more civilians towards the end of the sampling in the clusters, and we extrapolated this to the whole MDR population over 6 years, and we're able to find that that up to 30% of MDR-TB over the 6 years could be linked back to prisons. And this is an important finding because it offers the possibility for targeted interventions. So intervening in prisons would benefit the entire population, the civilian population, the general population, basically. So that's a bit the end of of this story in Georgia, and I hope managed to show you that the fitness cost of drug resistance in TB is heterogeneous. It actually depends on epistatic interactions between the strain genetic background, which lineage the bacteria is, what type of drug resistance mutation it has, and whether it has compensatory mutations or not. And together, these interactions will modulate the evolution of drug resistance and the fitness cost of the bacteria. And second, we were also able to find, to determine what is the genetic makeup of a very successful genotype, and what in general, what factors shape the MDR-TB epidemic in Georgia, but also in neighboring countries which have similar genotypes circulating and similar problems as incarceration. And with incarceration, we showed that how prisons can act as also breeding grounds for these highly successful genotypes that then go in the general population and then transmit very successfully. That being said, thank you so much for your attention. And I would like to thank the people involved in this study, the whole team in Georgia, of course, at the National Center for TB and Lung Diseases, Tanya and her postdoc that worked on all the models, and of course the group of Sebastian Genyer in which I work. Thank you.